Hello, guys. Hello, everybody. Welcome to our free GCSE biology stream. Hello, who have we got in? Post a little hello in the chat so I can see who we've got here. And we are going to crack on and get started. How are we doing, guys? Where is everybody? What's going on? Hello there, Hudder. This video is going to be about enzymes there, Gazelle. Hi, Yashi and Eva. Yashi, this is your second time with me this evening. What are you doing, man? What are you doing? Hello, Mathilde. Hello, Aisha. How are we doing, guys? How is everybody? It's lovely to have you all here. It's fantastic. There's no workbook for these free streams, Pranav. There's no workbook. Make some notes on a piece of paper if you want. We're going to be going through the 5th required practical, all about enzymes. Guys, sharing is caring. Send this link. Send the link to the live stream to your mates. It is free. So send it to as many people as you can. Let's get as many people as possible involved. Hello there, Sanjid, as well. Good to have you back. All right, guys. I put a little poll in the chat. Given that this is GCSE biology, who's ready for next Tuesday? Who's ready for next Tuesday? Is this predicted to come up on paper 1? Yes, Kenzie. It comes up so often. It's probably quite likely. One of those practicals that there's also almost certainly going to be something linked to rates affecting enzyme. Just half an hour, Aisha. We're going to be done by 8 p.m. It's just going to be half an hour. We're going to go through this practical. We're going to talk about all those different factors affecting enzyme action. What is next Tuesday, Yashi? Next Tuesday is biology paper 1. It is the first GCSE exam. Is this the last live stream? This isn't the last live stream, Gazelle. It is next week. Next week is going to be the cram course. So if you guys haven't signed up for the cram course, check the link out down below. It is going to be the cram course the night before your exam. So make sure you are there. I mean, English Lit might be on Tuesday as well, Aisha. I don't know when the English exams are, but I know that the GCSE biology exam is going to be next Tuesday. And then we're going to have so the cram course next Monday. And then the Monday after that, we're going to be back with more free streams up until paper 2. All right. All right, guys. Are we ready to start? Hello, Eva. Hello, S. And Oliver. Good to have you in. And Jumi as well. All right. Let's crack on and make a start. Let's begin. So we're talking enzymes today, guys. Your first exam is tomorrow, Lily. Good luck for tomorrow. Good luck. I hope you smash it. Alongside thinking about enzymes, though, we also need to think about the food tests as well. And we're just going to do a really, really quick recap of those food tests. And hopefully we all know those food tests really well and we can get those out of the way. Is this for foundation or combined? This would be for both. This would be higher and foundation, combined and triple as well. Hello, Artin. Good to have you in. Hello there, Chi, as well. So then we're going to be investigating how different factors affect the rate of enzyme-controlled reactions. So let's crack on. Let's go through our kind of food tests here. Sorry, no, we're thinking about enzymes. We're thinking about the role of different enzymes. We're thinking about how different enzymes are breaking down different substances. So we're starting with our product is glucose. Can we fill in the boxes? What is the enzyme that's going to be producing glucose? What is our enzyme that's going to be producing glucose? Fantastic there, Neve and Kenzie and Kay. This is going to be amylase. This is our amylase. And what is the substrate for amylase? The amylase is going to break something down. What's it going to break down to turn it into glucose? What is that amylase breaking down? It is starch. Exactly there, team. Very good. Very good. We are breaking down the starch into the glucose. All right, let's talk about proteins. What is the enzyme that's breaking down proteins then? What is our enzyme that is breaking down proteins? That is going to be our protease. Excelente, our protease breaking down those proteins. And what is the product of that protease, of the breaking down of those proteins? Good to have you in, Pamela. Good to have you in, Sal, as well. Great there, Neve and S. Fantastic, Daniel. Yeah, it is our amino acids. Amazing team, amazing, amazing. All right then, so our lipase then, our third and final enzyme. What is that lipase breaking down? Ooh, fancy Mathilde, mentioning trypsin and pepsin as examples of those proteases. It is breaking down the lipids. Very good, Sanjit. Very good, Aisha as well. We're breaking down the lipids. And what are we breaking down those lipids into? What are those lipids being broken down into? Very good, Yashi. Super quick there. Excellent, Jumi and Giovanni as well. It is those 3 fatty acids and the glycerol. So we've got our 3 fatty acids and glycerol. Fantastic, team. All right, let's talk about our food tests then. Let's talk about our food tests. So let's talk about how we might test for starch. What am I going to use? What reagent would I use to test for starch? This will be all exam boards there, Aman. This will be all exam boards. Very good there, casual viewer. And K, we are going to use iodine. And the iodine, what is going to be that starting colour of the iodine? What's going to be the starting colour of the iodine? What do we think there, team? Very good, Aisha. Brown or orange? Yeah, lovely. So we can just say orange there. And if we have that positive result, if it is positive, if starch is present, it is going to change colour. And what is it going to change colour into? What is it going to change colour into? It's going to change from orange to that kind of blue-black colour. Very good. Blue/black colour. Now, sometimes it might be necessary to say iodine solution. I think the Edexcel exam board, you have to be specific and say iodine solution. But iodine will usually just get you the mark. All right, let's go protein now. Let's talk about protein. What is our reagent to test for protein? Very good there, Mathilde. Super fast. Love that. And Pranav as well. Excellent. Protein is our Bioret reagent. Very good there, Kay and Rahana as well. So that is our Bioret reagent. And our starting colour for our Bioret reagent? What's our starting colour for Bioret reagent? What do we think? Great there, Kay and Yashi. That is blue. And great, Mathilde, giving our positive result. If it is positive, what is the colour change going to be? What is that positive colour change? Very good there, casual viewer. And Yusuf, it is going to change to lilac. Or we could just say purple, guys. We could just say purple. You're going to get the mark for saying purple. All right, let's go lipids next. Let's go lipids. Now, I reckon there's going to be a little bit of variation here. What would we say for lipids? Nicholas comes in with the ethanol. Kay saying ethanol and water. Very good. So ethanol plus water. Fantastic. Now, some of you might be thinking Sudan 3. Kwamnaz says Sudan 3. Now, Sudan 3 is another one that you could say, but you need to know the ethanol and water because in exam questions, they might tell you about the ethanol test or they might kind of give a description of the ethanol test and you need to know, they're testing for lipids, exactly. So what's the starting colour, I guess, let's say? What's the starting colour for the ethanol test? It is clear or colourless. Very good. Excellent there, Sanjid. And after, if we get a positive result, it is going to go from a clear to a white emulsion or cloudy. Very good, very good. So we're going to have a cloudy white emulsion. And that's why it's also called our emulsion test, a cloudy white emulsion. All right, I've run out of space. Let's go to Benedict's test. I'm slightly giving the answer away. All right, Benedict's test. What is the Benedict's test? What is the Benedict's reagent testing for? What is that Benedict's reagent testing for? Very good there, Nicholas, saying we're testing for sugars, or we could say glucose, reducing sugars. Aisha says, yeah, we're testing for sugar. Glucose being an example of that sugar. And the Benedict's reagent, what is our starting colour of our Benedict's reagent? What's the starting colour for the Benedict's reagent? So clear or colourless would be fine there, Kenzie. Blue, very good, team. Blue is our starting colour for the Benedict's reagent, and the colour is going to change to, what is it going to change to if the sugar is present? What's it going to change to if that sugar is present? like a brick red. We're going to go right the way through that traffic light. We're going to go green, yellow, orange, brick red, depending on how much sugar is present. Very good. Then 919X, it is changing to the brick red color. Brick red precipitate. It's going to form that solution that we can't see through as well. Could you get a 6 marker for a food test? Question. There has been one in the past, Chi. I don't think there's likely to be another one. I don't think it's likely to have a 6 marker on this, but it could happen. It has happened in the past. And then the Benedict's test, there's something else we need to do to the Benedict's reagent. What else do we need to do to the Benedict's reagent? What else would we need to do for this to speed up the rate of formation of the precipitate? Kenzie says heat. Pranav says heat as well. We are going to heat it. Sal giving a specific temperature, saying to 75. Yeah, I think it's good to give an example of a temperature. In the past, it has come up in exam questions where you have had to give a temperature, and like anything above 70 would be fine. Okay, so heat to 70 degrees Celsius. Yes, this is going to apply for IGCSE as well, there, casual viewer. So these food tests, although you may not get a question that is like specifically about the food test, we need to know these really, really well, because if we don't know them really, really well, then there's going to be other questions that we're going to have to apply this knowledge and understanding to. And if we don't know these food tests, that's going to make it a real challenge. All right, guys, let's move on. Hello there, Chai. I remember you from year 7 and year 8. I remember you. Okay, so investigating the rate of enzymes. Oh, we skipped a slide. There we go. Enzymes required practical. Our aim is to investigate the effect of pH on the rate of reaction of amylase. Now, there's a lot of different ways this practical could kind of be framed to you, could kind of be done in different ways. This is the way that you probably would have done it in school, but we might want to think about this slightly differently. So what we're going to do is we're going to use the enzyme amylase to break down starch at a range of pH values using iodine solution as an indicator for the reaction occurring. Now, the iodine solution is going to act as that indicator because it's going to show us whether or not the starch has changed color. Okay, it's going to change color depending on if the glucose is still present. Yes, this is for GCSE there, Abanit. So if the glucose has been broken down, if the reaction has finished, what will happen? So when the reaction has finished, what will happen to that iodine solution? When the reaction is finished, what will happen to the iodine solution? What do we think? What do we think? What would happen to that iodine solution if the reaction has finished? Is it going to change color? Aisha saying remains blue-black. Is it going to remain blue-black? It will no longer become blue-black. Yeah, it's going to remain orange. It's not going to change color. It's going to remain orange, and as a result, it won't change color. Excellent there, team. It's going to remain brown as well. Yeah, all that starch has been broken down. Excellent, Mathilde. And that means that there's no starch left to change the iodine, change the color of the iodine. So we can use the time it takes for that, can't we? The longer it takes for that iodine to not change color, the longer the reaction is taking, and so the slower the rate of the reaction. Remember, more time means smaller rate of reaction, means lower rate of reaction. So the amylase is an enzyme that digests the starch into sugar. The starch can be tested for easily using the iodine solution. And as we have done, as we've gone through our iodine solution there. So let's go through our practical method. What we're going to do is we're going to set up our spotting tile. And initially we're going to add the iodine to the spotting tile, and the spotting tile is going to have this kind of orange color because no starch has been added yet. We're going to then add the amylase and the buffer to a test tube, mix, then add the starch solution to start the add the starch solution and start the stopwatch. Now the buffer solution here, remember that's going to be our different pHs. So we're going to have different buffer solutions at different pH values. Before I mix these, what should I do before I mix these? What should I do to make sure that they are the same temperature? What should I do to make sure that they're the same temperature before I mix them? The buffer solution just keeps them at the same pH there, Gazelle. Yeah, great there, Yashi. I'm going to use that water bath. Fantastic, Sanjid, as well. Yeah, nice there, Lara. I'm going to let them equilibrate or acclimatize, equilibrate in a water bath so that they are both the same temperature before I mix them. And as soon as we mix them together, well, obviously the reaction is going to start straight away because we're going to have mixed the enzymes with the substrate. And so they're going to start breaking down. And then what we're going to do after 10 seconds, we're going to add a drop of solution to the iodine in the spotting tile. And we can see there, we can got 1, 2, 3, 4, 5, and then the 6th one there, the reaction is now complete because we've got a little bit of color change in number 5. And we're going to repeat every 10 seconds until the iodine doesn't change color. And we can see that here that we've got pH 5, takes 80 seconds. So 1, 2, 3, 4, 5, 6, 7. And then the 8th one is the one that doesn't change color. pH 9, 90 seconds, pH 14, no reaction. So based on this data, based on these results, what would you say is the optimum pH for amylase? It doesn't really matter, Yusuf. It could be 10 seconds, it could be 20 seconds, it could be 30 seconds. It doesn't really matter. The thing, we're going to talk about it in just a second though. What do we think the optimum pH would be? Neve says 5, Nicholas says 7, Eva says 7, 919X says 7, and so does Amina. Very good. pH 7, this is going to be our optimum. This is our optimum because it has the smallest time or the least amount of time. And if it has the smallest time, that means it's going to have the greatest rate. Because remember, our rate of reaction is our, our volume of product, or whatever you want to say, divided by the time. We put the time underneath. The optimum means the maximum or the best there, Adam, the fastest rate. So somebody asked a good question about, well, what does it matter? Why are we using every 10 seconds? At school, we did 30 seconds. Why does it matter? Well, the reason it matters is because we've said it takes 60 seconds for the reaction to be completed. But between 50 and 60, maybe it finished between then. Maybe it was 55 seconds. Maybe it was 57 seconds. Maybe it was 53 seconds. I don't know. As well as that, what if the optimum is somewhere between pH 7 and pH 5? What if it's somewhere between that? How could I determine whether the optimum pH was somewhere between 5 and 7? How could I find out if it was between the middle there? How could I determine that kind of better, more accurate point for that? Very good there, Kenzie. To determine that kind of point in the middle where the optimum pH would be, I perhaps need to use smaller intervals. So smaller intervals would then help to determine if the pH will determine what the optimum pH is. So it would make our final optimum more accurate. So it improves accuracy, let's say. Improves the accuracy. Hi there, Narika. Welcome. Improves the accuracy. People saying pH probe. Remember, guys, the pH probe is to measure the pH of the solution. We are trying to identify the optimum pH. So we're trying to find the pH at which the enzymes are working at their highest rate. All right. So when we think about an experiment, we need to identify our variables. What are our variables within this experiment? What is our independent variable? What is our independent variable? What if they ask this, but for temperature? Very good question, Aisha. Well, we would use a water bath to control the temperature, and we would have different water baths at different temperatures. And then we could, investigate it that way. Very good there, Lyal. Very good, Alfie, as well. Our pH is our independent variable. Our independent variable is what we change. And so our pH of the solution, or our pH buffer. What about our dependent variable then? What would we say for our dependent variable? What is the dependent variable in this investigation? This is the practical to test the amylase solution there, Adam. Very good. Yes. It's our rate of reaction. But what are we actually measuring? We're going to calculate the rate of reaction, but what are we actually measuring in this? Because we can't really measure a rate of reaction. We're going to measure the time. Very good, Neve. The time for what? So we're going to measure the time. The time taken. Are we measuring the time taken for the color to change or for the color to not change? So the time until the iodine solution, until it does not change color, until it stays the same. Stays the same color. Because when it stays the same color, that indicates that all of the glucose has been broken down. And so to convert that into rate of reaction, I'm then going to do our rate is equal to 1 divided by that time. 1 divided by the time would be our rate of reaction. What about our control variables then? The IV is the independent variable there, car comparisons. The dependent variable is, the DV is the dependent variable, okay? So Eva says heat or temperature, very good. Temperature would affect that rate of the enzymes. What else could we say? What else could we say? Sanjid says temperature as well. Lael says the volume, very good. So our volume of our enzymes, just our volume of enzymes, what else could affect it? What else could affect it? So we could say the volume of enzymes, the volume of the amylase. S says concentration, good. So we could say volume / concentration. Our volume of starch as well. Yeah, good. So our volume of enzymes / starch, and also our concentration of enzymes / starch as well. Is time a control variable? Bonus question. Is time a control variable for this investigation? Alfie says no. Amina says no. Eva says no. No, time is not a control variable. However, the time interval should really be a control variable. It wouldn't really matter that much, but the time interval would perhaps be a control variable. So we're going to measure it every 10 seconds, but the total time is what we're going to measure at the end, okay? Good. Let's have a little think about how we can keep these the same. So how would I control the temperature? How would I control the temperature? Great there, Kenzie. Yeah, very good. Yashi as well. It is a water bath. Great there, team. Very good. I love matcha. I really don't love matcha. Matcha's disgusting, guys. It just it tastes like grass. It tastes like agriculture. Will I be doing these for A-level biology? Potentially casual viewer. And then what do I mean by control experiment? What do I mean by the control experiment there, team? What do we think? What do we think? Not just repeatable, not removing the anomaly. Fantastic there, Giovanni. We're going to remove the enzymes. We want to show that it's the action of the enzyme that is causing the iodine to change color. One with no amylase, exactly. So one with no amylase. And that way it shows that the iodine doesn't just change color by itself, that the iodine is going to be changing because of the action of the enzymes. All right, how do I make my experiment better? How do I improve the precision of my experiment? We talked about how to improve our accuracy, use smaller pH intervals. How can I make it more precise? Matilde says taking repeats. Kumwan says repeat as well. Eva says repeat. But what else do I need to say, guys? I'm not going to get a mark for just saying repeat. I need to say repeat. And how do I get that mark? I'm going to say repeat and then do what? Very good. Yashi saying calculate a mean. Yes, repeat and calculate a mean. And we could also perhaps say removing, identifying, or removing / identifying anomalies as well. Fantastic there, Farhan and Kira too. Now, good. All right. We've got 5 minutes left, and I am going to give you a format that you can use to plan an investigation in your exams, okay? Because this type of exam question troubles people no end. So many students lose marks on these types of exam questions because they don't know how to phrase their answer, because they don't know how to do it, okay? And the really, really simple format for you to use is to just go through the variables. Variable, what you're going to do with it, next, okay? It doesn't really need to be like a recipe. You don't need to give your method as a recipe. What you're going to do is you're going to go through each variable. So the first point we're going to make in our answer is we're going to say the independent variable is, and in this case, pH, the pH. And what we're going to do is we're going to say how we're going to change the pH. So changed using how am I going to be changing the pH? What am I going to use to produce different pHs? It's not going to be 1 mark per bullet point. It's going to be marked as levels. And to get into that top level, to get 5 to 6 marks, what you're going to need to do is give a full description covering all of the variables and how to control, measure, or change all of those variables as well. And talk about how to improve the experiment too. Very good, Matcha and Narika. We're going to change the pH using different buffer solutions. After I've talked about my independent variable, I'm now going to talk about my dependent variable. So my next bullet point, I'm going to say the dependent variable is, and in this case, I'm going to say the time taken for the iodine to not change color. Now, how am I going to give a description as to how I'm going to measure this? How am I going to give a description of how to measure this? So the time taken for the iodine to not change color, what do we think there? We're going to use a stopwatch, very good. So thinking about mentioning our apparatus, so we're going to use a timer. We're also going to use our spotting tile. So I could maybe have like a little sub bullet point. We're going to have iodine solution on the spotting tiles. So the iodine solution on the spotting tiles. And what we're going to do is we're going to add the reaction mixture. We're going to add the reaction mixture every 10 seconds perhaps, or we could say 30 seconds. And then the iodine will change color. So the iodine will change color if the starch is still present. Very, very good. Okay, the spotting tile is the little plastic thing that would have had little dimples in it. And then we can talk about our control variables. So the control variables are, and we would mention all the ones that we have talked about. We could talk about temperature, and then we could talk about volume of enzyme / starch. Now, when we talk about temperature, how would I keep the temperature the same? Mentioning this in my method to investigate the pH. What would we say about the temperature? We're going to say using a water bath. Very good. So using a water bath, keeping that temperature the same. The volume in the volume of the enzyme or starch, how am I going to keep that the same? How would I keep that the same? Our volume of enzyme and starch, thinking about the apparatus that has been shown to us in the question. Using a measuring cylinder. So measured with the measuring cylinder. And then exactly as we have just done in our previous slides there, team, we're going to talk about how I could make my experiment more accurate and more precise as well. So we could talk about how to improve our accuracy. So to improve accuracy, How would I improve my accuracy there, guys? How would I improve my accuracy of our experiment? So really good there, Alfie. So to improve our accuracy there, team, we want to use smaller intervals because we want to know whether the optimum pH is pH 7 or maybe 7.5 or is it 6.5. We want to use smaller intervals. So use smaller intervals in pH. And then to improve our precision, running out of space at the bottom, to improve precision, I'm then going to I'm then going to repeat the experiment, and then I'm going to calculate a mean. Repeat and calculate a mean. And there we have it. 6 out of 6. 6 marks for your exam. Easy, easy, easy. And you can use a format like this for any of your describe a method questions, okay? You can use something like this to describe a method in your exams and you'll get 6 out of 6 every single time, as long as you hit every single one of these points. All right, guys, thank you so much. Thank you for joining. There's a few questions on the chat, so I'll come back to those in just a second. Now, next Monday is the GCSE cram course. So if you haven't signed up for that yet, check out the link below. Check out the link below to get signed up to the cram course next Monday. It is absolutely free as well. So it's absolutely free. It's going to be done by Laura. Laura does biology. If you don't follow her on TikTok, make sure you check her out as well. And then I'll see you the week after. Okay? And then I'll see you the week after. Because I'm going to do another free one the week after that. All right. Amazing. Get signed up to the cram course if you haven't already. Thank you so much for joining, guys. It's been a pleasure having you here. Amazing, amazing, amazing. Share the video. Share it with as many people as you can. Let's help everybody out. Let's get everybody grade 9s. Although, theoretically, everybody can't get grade 9s. So, let's just help everybody. All right then, teams. All right then, let's go. See you later. Thank you so much. All sciences, S. All sciences, they are the day before the exam. All right? Goodbye, all. Thank you, thank you, thank you, thank you. Kenzie, so I don't have to say things like first add 2 to 3 drops of iodine solution to a spotting tile. No, you don't need to say something like that. You'd need to say like have the iodine solution on the spotting tile because the iodine solution changes color when the starch is still present. That's what you'd have to say, but you don't need to give like really detailed descriptions like it's a recipe. Thank you so much, guys. Thank you so much. If you don't do AQA, I mean, the content, there's so much overlap in the content, Alfie. The problem is the other exam boards, different topics might be on different papers. So the papers that you're going to be doing next Tuesday might have completely different topics on it. That's the problem. Okay, Alfie? So if there is overlap with that, then yeah, it would probably be useful. So in 2 weeks' time, Aisha, we're going to go through some of the paper 2 practicals, all right? Yeah, exactly, Chi. The little summary, the summary's right there, Lara. The summary's right there. Everything you need to know right there. 2 weeks, Michael. 2 weeks. Keep an eye on my TikTok. I'm going to be posting on my TikTok to let you know, guys, to let you guys know when it is going to be. All right. Thank you so much. Goodbye. Goodbye. Goodbye.